Okay, so let's just, we're going to start at the beginning. I'm going to keep going back and forth with these clips so that you can see the relevant pieces, but we're going to be starting with just an overview here. So key events that happen in the light-dependent reactions are, one, photoactivation, and that's what you were seeing happening in that video previously, and we'll go into much more detail on that in just a second. And this is where the photons of light strike the accessory pigments in photosystems 1 and 2, and the energy that is absorbed results in the excitation of the electrons. So it takes the electrons from a lower energy level to a higher energy level. And this is the energy that we're basically absorbing from light that we're going to convert into chemical energy. Following that, photolysis occurs, and we'll look at that in detail. And that's when ultimately you saw that those electrons, they end up getting ejected from the chlorophyll molecules in the reaction center. And when they're ejected, they have to be replaced. Otherwise, we can't continue absorbing more light. So they need to constantly be replaced. And the source of that is water molecules. So a water molecule is split at photosystem 2. And this results in molecular oxygen, which we know is a waste product. But it also produces protons to help us build up that proton motive force, or the electron gradient that we talked about in cellular respiration. And it also produces 2 electrons that are going to replace the 2 that were lost from the chlorophyll A molecules at the reaction center of photosystem 1. As a result, this will then continue this process of replacing and taking and replacing and taking, so that we can continue to harness that energy from the sunlight. And then those electrons, where do they go? Well, you guys are familiar with redox reactions. So redox reactions are going to move those high energy electrons, and they're going to use all the energy out of those electrons so that they can do, you guessed it, pump hydrogen ions from the stroma into the lumen, which is a small space just like the inner membrane space. And we're just going to build up that proton gradient so that we can then use that to make ATP later. And that ATP is not necessarily for cellular ATP, that's for the Calvin cycle. And then finally, ATP is going to be made through the same function that we saw in cellular respiration. And we're also going to reduce NADP+. Now, this is just like the NAD molecules, NAD positive plus molecules that we saw in cellular respiration, only there's a P in it, right? So NADP+ gets reduced to NADPH. So again, instead of oxidizing them at the end, we're going to reduce them. And again, why do we want to do that? Because we want to move electrons from one place to another, and we need electron carriers to do that. So let's look at the structure of the chloroplast and then photoactivation in some detail. So the chloroplast, as you can see here, it has a double membrane. Remember, we've got the outer membrane, and then you've got the inner membrane. And then, unlike the mitochondria, this inner membrane does not form the cristae, and it does not increase the surface area, because where we need the reactions to take place is in these thylakoids. And so that's why you see this membrane as in just 2 solid membranes surrounding each other. So this yellow space in here, we would normally refer to this yellow space as like a matrix, but here it's called the stroma. Okay, and this is filled with the aqueous fluid, and the Calvin cycle, or the light-independent reactions, take place here in the stroma. And then you've got your thylakoid. So a thylakoid is going to be this structure here that's surrounded by a membrane. And one of these is the thylakoid. A stack of them is called a granum, and then all of them, when you have multiple stacks, that's grana. And they're connected, and they're interconnected by this inner membrane system. And inside of this, you can see that we've created this small space in the lumen of the thylakoid, and this is where we're going to build up that hydrogen ion, or that proton gradient here, so that inside it's more concentrated than in the stroma, so that we can then rush the hydrogen out, and then ATP can be formed in here to be used in the light-independent reactions. So if we look at zooming in on the thylakoid membrane, and you guys will get a chance to draw and label this a little bit later, but if you look here, you'll see that you've got just like a membrane like we're used to seeing, with the phospholipids on either side here, with the nonpolar region and the polar region. And then here is the first photosystem, which is photosystem 2. And photosystem 2 is consisting of a bunch of different accessory pigments, and the one that we're going to focus on are the 2 chlorophyll A molecules that serve as the reaction center, because that's where we're going to end up taking those high energy electrons and moving them down this electron transport chain that you see going along here. And then we've got, remember Q from cellular respiration, we've got plastiquinone here. And plastiquinone, again, lipid soluble. And it starts out on the stroma side, and it's going to move over to the lumen side here. And when it does, it's going to bring with it some hydrogen ions. And then it's going to deliver those electrons over here to cytochrome B6F, and this one is a protein complex, just like the protein complexes in cellular respiration, and it's going to pump hydrogen ions to the lumen from the stroma, and in doing so, it's going to use the energy from the electrons through redox reactions to be able to fuel that. The low energy electrons will then get passed on to plasticyanine. Again, this is not lipid soluble, so this is going to be on the lumen side, and it's going to deliver those low energy electrons to the photosystem 1 to replace the ones that were lost there. And you can see that we've got light activation here, we've got light activation here, and then we're going to talk about the water splitting complex here, which is going to replace those electrons. And I'll pause for now, and we'll get into this stuff a little bit later, but let's go ahead and move on. So what happens during photoactivation? So photons of light, so here's the sun, here's our photons. Photons of light are going to hit these accessory pigment molecules here. And this is photosystem 2 because that's where we're starting, but it also is going to be doing the exact same thing in photosystem 1. And again, only certain wavelengths of light are absorbed, primarily in the range of 400 and 700, so like 450, so on and so forth. And that's going to be the blue, right? And then also in the range of the 700 is going to be your red. And these light photons, the wavelengths of these light are going to be absorbed, and then they're going to cause electrons to jump up, almost like if you're doing the wave. If you do the wave in like a stadium, right? And so energy hits and your arms go up, but you don't pass your arms to somebody else. Your arms come back down. But when they come back down, what do you notice happening to the person next to you? They raise their arms up, right? And this is kind of how the energy, it fluoresces from one pigment molecule to the other. They maintain their electrons all the way until it gets to the reaction center. And in the reaction center, there are going to be 2 chlorophyll A molecules here. But when those electrons go up, they are captured by the primary acceptor, which is going to then deliver them to plastiquinone. And this plastiquinone then is going to be the carrier that's going to begin basically the initiation of that electron transport chain. Okay, now what happens here? So we are now, if you see here, this is where we've gotten to, we are now missing these 2 electrons here. And that's problematic because if I try to start this again and I start fluorescing and the enzyme, the electrons keep going up and down and up and down and up and down and up and down, and then they get to the reaction center and there are no electrons to excite, then we've just stopped. So that's the end of photosynthesis if we can't find a way to replace these 2 electrons that are missing here. Okay, and so that's where we're going to end up going to the next bit here. So this is just explaining here, Q starts out on the stroma side. Remember, it's going to start on the stroma. It's going to pick up 2 hydrogen ions with it, and it's going to take them down to this cytochrome B6F complex here. And it's going to deliver those protons down into the lumen so we can continue building up that proton motive force. So what is photolysis? So if you look up here, you can see photosystem 1. Here's the light that's triggering. You can see that the electrons are fluorescing and passing that energy all the way to the reaction center, and here's our reaction center, okay? So at the reaction center, again, the electrons get excited, and when they get excited, they get stolen or they get ejected onto this plastiquinone molecule, or Q. And it's going to take it, and it's also going to pick up some hydrogens up here, and notice that it travels down here. It's going to drop hydrogens off into the lumen, and notice this is the small lumen space here. And then it's going to deliver those electrons to cytochrome here. So it's going to, here's the primary acceptor, passes it to plastiquinone. This is what's moving back and forth between them. And then it gets to the cytochrome B6F here. And that's where pumping of hydrogen ions. So you'll notice here plastiquinone hydrogens are coming across with it. Here cytochrome B6F hydrogens are coming across with it into this to build up that tension and that gradient in here. So when we do that, we've got to figure out a way to replace these 2 electrons. And the way we do it is through an enzyme complex called the water splitting complex. And it basically takes water molecules and it splits them into molecular oxygen and protons. And these protons we need because they're just going to continue building up this force in here to be used later on with ATP synthase down here, right? But the oxygen, we know we don't need. That oxygen just turns into molecular oxygen, and then it diffuses out of the cells. And that is why one of the waste products of photosynthesis is oxygen, and why we can use that to be able to measure the rate of photosynthesis, how much oxygen is actually being produced. And it's all relevant to this particular complex here. So the oxygen is going to diffuse out, and then the two electrons, you'll notice if I have two hydrogens, I also had to have two electrons connected to those two hydrogens. And those electrons are going to replace these two here, and they're low energy, and they're going to wait until the next photon activates another one of these molecules, pigment molecules, and it's going to spread that then, and then these will be excited, stolen by the primary acceptor, passed to plastoquinone. It's going to take it across, same thing over and over again, and this just keeps repeating. I'll let you go ahead and look at some of these notes here. Notice here is the equation. You can also see this equation as 2 water molecules and 1 oxygen, not half of one, and then 4 and 4, which is acceptable. And then again, like I said, oxygen is a waste product. The electrons are used to replace the 2 lost, and then the protons are used to help build up that proton motive force. And it's very similar to what we saw in cellular respiration. So we're going to watch a little more specific rendering of this to see if you can follow along with what we just talked about. This is going to be another animation of photoactivation.
Photosynthesis occurs in the chloroplasts of plants and algae. The process is also found in single-cell organisms, such as cyanobacteria, that do not have chloroplasts.
So remember when we talk about, we know cyanobacteria is one of the photosynthetic organisms, but remember it doesn't have membrane-bound organelles because it is a prokaryote. So only the eukaryotes, which would be algae and plants, are going to have the chloroplasts in it because chloroplasts are obviously membrane-bound. In fact, they have a double membrane.
Like its mitochondrial counterpart, the chloroplast electron transport chain consists of several proteins.
Just really quick, notice that this is going to be where we have the lumen here, and then the outside space here where the ATP is produced and the NADPH is going to be produced as well. This is going to be the stroma area.
Protein complexes and mobile electron carriers. First, a photon of light hits a chlorophyll molecule surrounding the photosystem II complex. This creates resonance energy that is transferred through neighboring chlorophyll molecules. When this energy reaches the reaction center embedded in photosystem II, an electron is released. The reaction center chlorophyll contains electrons that can be transferred when excited. 1 photon is needed to excite each of the electrons in this chlorophyll. Once excited, 2 electrons are transferred to plastoquinone QB, the first mobile carrier.
So before, you have to have 2 electrons, which is why we talk about the 2 chlorophyll A molecules. Each chlorophyll A molecule will eject 1 electron, but you need both of them in order to have plastoquinone QB move.
In addition to the 2 electrons, QB also picks up 2 protons from the stroma. The 2 electrons lost from photosystem II are replaced by the splitting of water molecules.
Notice that the water splitting complex is in the thylakoid lumen, and where we just saw it picking up the plastoquinone picking up the hydrogen ions, that is going to be in the stroma.
Water splitting also releases hydrogen ions into the lumen. This contributes to a hydrogen ion gradient, similar to the one created by mitochondrial electron transport. After 2 water molecules have been split, 1 molecule of molecular oxygen is created. Plastoquinone QB then transfers the 2 electrons to the cytochrome B6F complex. The 2 protons it picked up are released into the lumen.